Good morning once again. Our next topic is cell motility by cilia and flagella, their organization and functions. What is cilia and flagella? The cilia and flagella are microscopic, contractile and filamentous processes of the cytoplasm which create food currents, act as sensory organs and perform many mechanical functions of the cell. The cilia and flagella are mainly projections from the cell, and the main difference between them are by their length. Morphologically and physiologically, the cilia and flagella are identical structures, but even then both can be distinguished by from each by their number, size and functions. The flagella occur at one end of the cell, while the cilia may occur throughout the surface of the cell. This is the picture of cilia. Cilia is a hairy-like structure that is seen here, and flagella is a long thread-like structure. Next is the distribution of the cilia and flagella. The flagella occur in the protozoans of the class Flagellata, choanocyte cells of the sponges, spermatozoa of the metazoa, and among plants in the algae and gamete cells. The cilia occur in the protozoans of the class Ciliata and members of other classes and ciliated epithelium of the metazoa. The cilia may occur on external body surface and may help in the locomotion of such animals as the larvae of certain platyhelminthes, nematines, echinodermata, mollusca and annelida. Next is the structure of cilia and flagella. The ciliary apparatus is composed of the shaft or cilium, which is a slender cylindroid process that projects from the free surface of the cell. Next is the basal body or granule, a centriole-like cellular organelle from which the cilium originates. Next is the ciliary rootlets in some cells like fine fibrils. The cilia and flagella are extremely delicate, permanently formed thread-like extension of cytoplasm, and their thickness is often at limit of the resolving power of the light microscope. Next, this is the microscopic structure of the cilia, and this is the ciliary membrane, ciliary axoneme, basal body. This is the plasma membrane of the cell, and this is the passage through which the fluid flow or nutrients for the cilia takes place. Next is the origin of cilia. The newly formed basal bodies become aligned in rows beneath the apical plasma membrane, and each basal body may then produce satellite from the side, a root from its base and a cilium from its apex. The formation of the cilia from the basal body is started by the formation of a vesicle-like structure of the cytoplasm towards the distal end of the basal bodies. The wall of the vesicle are invaginated due to rapid growth of ciliary shaft. The walls of the vesicles are temporary and are replaced by the new and permanent ciliary sheaths. Next is the ultrastructure of cilia and flagella. A eukaryotic cilium or flagellum is composed of mainly three major parts: a central axoneme or shaft, the surrounding plasma membrane, the interposed cytoplasmic matrix. First is the ciliary membrane. Though the ciliary membrane is physically continuous with the plasma membrane of the cell, but it contains far less amount of proteins than the latter. A ciliary necklace is found at the region in the cilium where microtubules and the basal bodies make contact with the membrane. They have mainly two functions. It may position the underlying basal body from which the cilium is originated, and the second is it may help in the differentiation of ciliary membrane, that is, the rings of the particles may retain proteins that would otherwise diffuse out and be incorporated into the ciliary membrane. The second one is the matrix. The bounded space of the cilium contains a watery substance known as matrix. In the ciliary matrix, they are embedded with 11 microtubules of the axoneme and the other interconnecting proteins. The third is the axoneme. The axial basic microtubular structure of cilia and flagella is called the axoneme. It is the essential motile element of these organelles. The axoneme is about 0.2 to 10 μm in diameter and may range from a few microns to 1 to 2 mm in length. The cilia may be thicker at the base and may become thinner gradually along the length. In the center of the axoneme are two singlet microtubules or fibrils that run length of the cilia. Each of the central microtubules is composed of 13 protofilaments. The central fibrils each has a wall of 6 nanometer thick and are located 35 nanometers away from each other. Both central fibrils are connected by a bridge and are enclosed in a common central sheath. A plane perpendicular to line joining the two central tubules divide the axoneme into a right and a left symmetrical half. It is generally accepted that the plane of the ciliary bead is perpendicular to this plane of symmetry. 9 doublet microtubules, each with 36 nanometers in diameter, surround the central sheath. They remain separated from each other by a distance of 20 nanometer and from the ciliary membrane by a distance of 25 nanometer. Each peripheral doublet consists of 2 microtubules or subfibers. They are 18 to 25 nanometer in diameter. One is small, that is A, and complete. They are very completed, and they are having a 13 protofilaments of tubulin and lying closer to the axis. The other subfiber B, that is B, is larger and incomplete in structure, having only 11 protofilaments. The B subfiber lacks the wall adjacent to A subfiber and is skewed at about 10 degrees away from the axis. The other associated structures of the doublets are dynein arms. The dynein arms are the extending from each subfiber A. There are two dynein arms, an outer arm and an inner arm, that are oriented in the same directions in all microtubules. Next is the nexin links. Their adjacent doublets are joined or linked by peripheral interdoublet or nexin links. The nexin links have a periodicity of 86 nanometer. Nexin links extend from a tubule of one doublet to B tubule of adjacent doublet. The third is the radial spokes. There are mainly 36. They are of length 36 nanometer long, radial bridges or links between the A subfiber and the sheath containing the central microtubule. These spokes terminate in a dense knob or head, which may have a fork-like structure. This is the structure of the axoneme. Here we can see this is the plasma membrane, and these are the dynein arms, and this is the nexin. The inner sheath, radial spokes, B tubule, A tubule, and B tubule, like that's doublet. They are the doublet microtubule, A tubule and B tubule, central microtubule, and all in this figure. Next is the motility by the cilia and flagella. The cilia and the flagella serve many purposes, and their movement propel the organism. The cilia are contractile structure, and in them two types of rhythms known as metachronic and isochronic or synchronous rhythms produce the waves of contractions in the cilia. The metachronic type of rhythm in the cilia of a cilia have a row beat one after the another, while in the synchronous or the isochronic rhythm have the cilia of a row beat simultaneously. The beating of the cilia or flagella is caused by intraciliary excitation, which is followed by a interciliary conduction. The movement of the cilia may be under nervous or cytoplasmic control. There are mainly 4 types of ciliary movements. They are pendulous ciliary movement, unciform ciliary movement, infundibuliform ciliary movement, and undulant movement. The pendulous ciliary movement. The pendulous type of ciliary movement is carried out in a single plane. It occurs in the ciliated protozoans which have rigid cilia. In such cases, the movement of the cilia is carried out by a flexion at its base. The next is a unciform ciliary movement. The unciform is the meaning is hook-like. Ciliary movements occurs commonly in the metazoan cells. In such type of movement, when the cilia contracts, it becomes double and acquires a hook shape. Next is the infundibuliform ciliary movement. The infundibuliform ciliary movement occurs due to the rotatory movement of the cilium and flagella. In this case, the cilium or flagellum is passed through three mutually perpendicular planes in the space and makes conical or funnel shape like that. The undulant movement. The undulant movement is the characteristic of the flagellum. In undulant movement, the waves of the contraction proceed from the site of implantation and pass to the border. Next, each beat of the cilium or flagellum involves the same pattern of microtubule movement. Each cilium moves with a whip-like motion and its beat may be divided into two phases. The fast effective stroke or forward active stroke or power stroke, in which the cilium is fully extended and beating against the surrounding liquid. The next is the slow process, that is recovery stroke, in which the cilium returns to its original state or position with an unrolling movement that minimizes viscous drag. In the case of flagellar movement or locomotion, there occurs as either planar waves or like beating or three-dimensional waves. All these three forms of flagellar movement consist of contraction waves that pass either from the base to the tip of the flagellum or in the reverse direction to produce forward or backward movement. The picture shows the movement of cilium. This is the power stroke, and this is the recovery stroke. This is the movement of flagellum. Next is the type of flagellar arrangement. This is polar or monotrichous, that is single flagellum at one pole. Lopotrichous, tuft or flagella at one pole. Amphitrichous, a flagella at both poles, and peritrichous, a flagella all over the cell, and amphilopotrichous, a tuft of flagella at both ends. It is well established now that the ciliary movement is generated by the microtubules and the associated structures of the flagellum. It was shown that the cells free can be used to move by adding any energy sources such as ATP. Even broad pieces of cilia or isolated axonium itself continue to beat, suggesting the role of microtubules in the movement. The contractile axostyle of some microorganisms such as Metamonadida, a dinoflagellate that lives in the gut of termite, is another example of the microtubule mediated motile process. The other functions of the cilia and flagella are mainly they helps in the movement, helps in the locomotion of the cell or the organism. The cilia create food currents in the lower aquatic animals. In the respiratory tract, the ciliary movements help in the elimination of the solid particles from it. The eggs of the amphibians and mammals are driven out from the oviduct by the aid of fibratile cilia of the latter. So that's all about my topic. Hope you all understood my topic, and thank you and have a nice day.